Welcome to the second episode of the Everything You Don't Need to Know series, where we take something simple and make it complicated. This episode is about the cell cycle, meiosis. If you haven't, I highly suggest watching our mitosis video first to be able to understand this one. My name is Camila Pimentel, and I'll be your guide today. Meiosis is a two-part process that produces four haploid daughter cells. Haploid means that they don't have chromosome pairs. They have chromosome units. They have 23 units of chromosomes at the end of the process. This is the type of cell division that enables the formation of eggs and spermatozoids. It also allows for genetic variability through a process called crossover. Entry into meiosis 1 depends on mCDK activation. Meiosis is essentially mitosis repeated twice. It begins with a cell and interphase. To review from the previous video, interphase has G1, S, G2. The G's are for growth and S is for synthesis. After G2, we have meiosis 1 that just like mitosis, it has karyokinesis and or nuclear division, or cytokinesis or cytoplasmatic division. G1 is where cells become committed to continuing the cell cycle or staying arrested in G0. There, proteins are built, energy reserves accumulate, DNA building blocks are produced, and the cell has 46 chromosomes. S is where cells replicate their DNA and centrosomes. The cell now has 92 chromatids. G2 is where cells rebuild their energy reserves and build more proteins and the cytoskeleton dismantle. Now, this picture is of meiosis of my this picture is of mitosis, sorry. So it only shows one daughter cell plus the original cell, each with 46 chromosomes. Meiosis one's outcome is similar, but not quite. Just like mitosis, meiosis is divided into prophase, prometaphase, metaphase, anaphase, telophase, and diakinesis. However, there are extra steps in prophase 1. Prophase 1 has leptotene, where the duplicated sister chromatids first condense into distinct thread-like structures. Homologous chromosome pairing also occurs during leptotene, in which the homolog axes are aligned with each other roughly 400 nanometers apart. In zygotine, homologs are brought even close together to a distance of about 100 nanometers in a process called synapsis. An array of protein filaments called the synaptonemal complex is assembled between the homologs. Pachytene is the stage at which the synaptonemal complex is complete, and homologs are tightly linked along their entire lengths. The chromosomes become highly condensed in diplotene, and crossovers become apparent as chiasmata, as you can see in the red circle. Crossovers is where homologous chromosomes, not sister chromatids, exchange part of their DNA. This causes enhanced genetic variability since a chromosome that originally came from the father can have a part that came from the mother, and so on. Non-crossover is when, instead of replacing the DNA, a DNA repair event happens. Each pair of homologs has at least one crossover, and that crossover tends to be distributed far apart from each other on the chromosome. Sister chromatid cohesion is completely absent at the chiasmata. Cohesion proteins concentrations are reduced along chromatid arms and are lost at the chiasmata in late prophase. This partial loss of cohesin depends on condensin. Condensin is thought to recruit the kinase PLK, which then phosphorylates cohesin. Diakinesis. Here the nuclear membrane starts to break down. The nucleolus disappears and the crossovers are done with. Double strand breaks do not occur randomly along the chromosomes, but are focused on preferred sites or recombination hotspots that are generally found in chromatin that is relatively accessible to proteins. Recombination is thought to occur in the chromatin loops away from the homolog axis. Meiotic recombination begins when both strands of the DNA double helix are cleaved at many locations by the enzyme SPO11. The exposed DNA 5 ends are trimmed back or resected, resulting in single stranded 3 ends overhangs that are then coated by recombinases called RAD51 and DMC1. The resulting protein DNA filaments, called strand transfer complexes, have the ability to invade another double stranded DNA and scan it for complementary regions of DNA sequence. In meiosis, a single strand from one homolog invades a chromatid of the other homolog. Recombination between identical sequences on sister chromatids is suppressed. On locating a homologous sequence, the invading filament pairs up with the complementary strand, displacing the other strand of the DNA duplex to form a single stranded displacement loop known as a D loop. Further DNA synthesis and ligation fills in the gaps, and the final result is a DNA structure called a double holiday junction. In prophase 1, the nucleolus disappears, centrosomes move, chromatids condense, homologous pairs pair up, and crossing over occurs. In prometaphase 1, the nuclear envelope disappears and centrosomes move to opposite poles of the cell. In metaphase 1, the homologous pairs align in the equatorial plane. Homolog pairs become bioriented on the first meiotic spindle. In anaphase 1, the chromosomes are pulled towards the poles, and the cell elongates. Spindles, checkpoints, system delays sister chromatid separation in response to defects in chromosome attachment to the spindle. To learn more about the spindle checkpoint, view the mitosis video. Centromeric Rec8 is protected from cleavage in meiosis 1 by a protein SGO1, making it so sister chromatids stay together, attached at the centromere. In phase 1, chromatin relaxes, the nuclear envelope reforms, and the nucleolus reappears. In diakinesis, the ring of actin filaments contracts, the cleavage furrow forms, and the cell cleaves into two halves. And then we get into meiosis too. Something important is that we don't have interface in humans in between meiosis 1 and meiosis 2. Thus, chromosomes will not replicate. Instead of sister chromatids, what's being separated here are the homologous pairs. We begin with two cells that are diploid, the chromosomes are in pairs with 46 chromosomes, and end up with four cells that have 23 chromosomes each. In prophase 2, chromosomes condense. In prometaphase, the nuclear envelope disappears. In metaphase 2, they organize in the equatorial plane. In anaphase 2, they get pulled to opposite poles. SGO1 disappears from the cell in the second meiotic division, thus exposing Rec8 to cleavage in anaphase 2. Telophase, nuclear envelope and nucleolus reforms, and diakinesis, the cytoplasm separates. And I want to view this image again after explaining all of that. Notice how, due to crossover, some chromosomes have mixed colors, or they are mixed with other chromosomes that ended up in other cells. Notice how we go from a cell exiting interface with 92 chromosomes and ending up with just 23 in four daughter cells. CDK1 partial inactivation is responsible for the inhibition of DNA replication during the second meiotic division. When Errors in meiotic segregation happen. We can have aneuploidy, where we have an abnormal number of chromosomes in cells. When these sexual cells form a zygote, they can carry over the errors and monosomies, and triploidies happen. Aneuploidy zygotes generally fail to survive, and meiotic segregation errors account for half of the spontaneous abortions in the first trimester of pregnancy. An example of a viable fetus with aneuploidy derived from an error from meiotic segregation is trisomy 21 or Down syndrome, where the 21st pair of chromosomes is not a pair but a triplet. I will talk more about meiotic segregation disorders or chromosomal disorders in another video. How do they happen? Let's say in meiosis 2, instead of dividing all the chromosomes by half, one pair of chromosomes remains and passes on to one daughter cell. This causes one daughter cell to have 47 chromosomes and the other will have 45. If these cells merge with other sexual cells and form a zygote, we'll have a trisomy and a monosomy, respectively. Recombination Recombination checkpoint system. This system depends on components of the same DNA damage response system that blocks the mitotic cell cycle in response to a double strand break or other abnormalities in DNA structure. Recombination defects block entry into meiosis 1 by preventing the activation of MCDK complexes. This one was a shorter video compared to what I want to do with the Everything You Don't Need to Know series because I'm separating gametogenesis from meiosis. Also, many aspects were already discussed on the mitosis extended version. Next week, we will have gametogenesis and we'll say bye bye to the precious 300-page book I've been reading for this. This is the book. The Cell Cycle Principles of Control by David O. Morgan. Um, as I said, the link is still down. So to access it, you have to go through the Wayback Machine and input the link that I will give in the description. I will give a shout out to myself for the drawings, editing, script, voiceover. Ivana Pimentel for script clarity, my family and dogs moral support, and you for watching this video and making it to this point. I want to give you a very, very good thank you and ask you to subscribe, like the video, and comment if you are able. Thank you very much and see you next time.